Good day to each and everyone. My name is Zeruel Bidalgo Jr. and this is my report about plant sensor systems, signals and responses for our subject Science 208 Biological Sciences under Dr. Genevieve Pipito. Plants have sophisticated information processing systems and can respond to several environmental cues. The stem of this plant, for example, senses the pull of gravity and is growing away from it. So the learning competencies for this topic to compare and contrast the following processes in plants, the chemical and nervous control, immune systems, and sensory and motor mechanisms. Now I made an organizational chart for us to give an overview about my topic for today. So plants sense and respond to a variety of environmental factors, starting with signal processing in plants as it relates to how plants respond to light, gravity, wind, and touch via directional movements, that is tropism, and via developmental changes. The signal processing in plants, then exploring the hormones involved in youth, maturity, and aging, and seeing how plants defend against pathogens and herbivores. There are three-step process by which plants gather, process, and respond to environmental stimuli. Step 1 is that the plasma membrane receptor protein changes its shape in response to the signal. Step 2, a signal transduction pathway transduces the signal, often a hormone, into an intracellular signal. Step 3, the intracellular signal triggers a response, changes in transcription of target genes, the activity of specific enzymes or transport proteins, or other changes in cell activity. So I am going to show to you an illustration or a visual representation about the steps in information processing that occurs in plants. So 1, there must be an introduction, or a signal must be introduced to a to an organism, specifically the plant. So the sensory cell perceives the external stimulus and transduces the information to an internal signal. So there is a change happening. And then when the signal enters the cell, there is now the so-called cell-cell signal or cell-to-cell signal. It is released by the sensory cell that travels throughout the body. Those cell-cell signal will go to the target cells because there must be a specific response. What part of the plant will respond from the external stimulus? So how do cells receive and process an external signal? The signal from the environment are usually detected by proteins specialized for that function. The signal receptor proteins change shape. So it's like change of shape in response to an environmental stimulus, such as what are those environmental stimulus we have? The struck by a particular wavelength of light, and next we have having pressure applied or binding to a particular type of molecule. When a signal receptor changes shape in response to a stimulus, the information changes form from an external to intracellular signal. This is to accommodate the size of the cells. Later you will know what made this external signal into intracellular signal. So what happens, like, is there a conversion happening? So what makes them change? The intracellular signal primes the sensory cell for action. We are familiar with hormones. It is an organic compound that is produced in small amounts in one part of a plant and transported to target cells, where it causes a physiological response. For example, a hormone produced in roots may cause stomata in leaves to close in response to dry soil conditions. If there is summer or if the condition of the soil is dry, the stomata in leaves must close to avoid dehydration of the plant. Now, some signal receptors exist inside the cell where they respond to signaling molecules that readily diffuse through the plasma membrane. However, most signal receptors are located in the plasma membrane where they respond to external stimuli and bind to signaling molecules that cannot or do not cross the membrane. If the receptor resides inside the cell, the information carried by the signal if the receptor resides inside the cell, the information carried by the signal is processed directly without any intermediate step. If the receptor is located in the plasma membrane, the subsequent process is called signal transduction. So, the thing that I have said earlier about the external stimulus changing into intracellular signal to accommodate its function inside the cell, so we have this process called the signal transduction. Transduction comes from the root word transduce, which means to convert from one form to another. So there is a change of its form. The signal transduction begins with the stimulation of a receptor protein in the plasma membrane. Signal transduction pathway often involve two fundamental components. We have the phosphorylation cascades and the second messengers. What are these? But before that, I am going to show to you a visual presentation about the signal transduction. The signal transduction, when the signal arrives, the receptor protein changes in response to the signal. This is now already the signal transduction. And then third, this receptor protein is located in the plasma membrane. So this is responsible for the signal to change. This is the reason the receptor protein is the one that will change the signal or the external stimuli into intracellular signals. What happened? So the receptor or associated protein catalyzes phosphorylation reaction. So in this one signal transduction, there are two processes happening here: the phosphorylation cascade and the second messenger. The phosphorylation activates proteins involved in signal transduction cascades, causing them to phosphorylate and activate a different set of proteins, which in turn catalyze the phosphorylation and activation of still other proteins. So in phosphorylation cascade, the ATP will change into ADP, and the process will go on um, until it reaches to a desired output. And then there is a release of second messenger. There are a release of ions. The phosphorylated proteins or second messengers initiate the response. So what happened? The second messengers are produced when receptor proteins trigger the production of intracellular signals or their release from storage areas. So we all know that calcium ions stored in the vacuole, ER, or cell wall are one of several ions or molecules that function as the second messenger in plants. The process by which DNA is copied to RNA is called the transcription, and that by which RNA is used to produce proteins is called the translation. So as you can see here, the ions activate or repress the transcription, or they will activate or repress the translation, and then they can change the ion flow through channel or pump. So it starts with a signal transduction, change of its form from signal, then there is a change of signal to create some actions inside the cell. So that's what is happening and during the information processing in plants. So now is we have the blue light. We are going to discuss about the three lights here: the blue light, the red light, and the far red light. The blue light is the phototropic response. I think you are familiar with Charles Darwin. In his San Francis, they experimented phototropism of grass seedlings. So as you can see here, we have the coleoptile. It is a modified leaf that forms a sheath protecting the emerging shoots of young grasses. We all know that Charles Darwin is famous for its origin of the species, and theory of evolution and his understanding of the process of natural selection. Also, they also studied about the behavior of plants. The blue light is a phototropic response. So we all know that light is made up of wide array of wavelengths, but plants sense and responds to only few. We are familiar with the Roig beam and light and the visible light, which is only a short portion of wide array of wavelengths of light. We have the visible light, and then this visible light is composed of colors. We are familiar with the Roig beam acronym for red, orange, yellow, green, blue, indigo, and violet. So now I have a photo here. There's a candle or a light. There's a fire there or that represents the light. There is a shoot there that bends. This shoot is bending toward the full spectrum of light. We still do not know what makes this plant respond to light, but we will later know what light is responsible for the bending. So the bending response was due to the elongation of cells on the shaded side of the plant. Directed movement and response to light is called the phototropism. So we have a candle here, but Darwins, they use paraffin lamps or other sources of artificial light in their original experiments. So in this picture, we have different kinds of coleoptile. They are colored based on what kind of light they are exposed to. So we have 5 here. In yellow, it doesn't bend. In green, it doesn't bend, but there is a small oblique direction as it grows. Orange and red, all of the 4 color mentioned is not showing some bends as compared to the blue light. It means that the blue light is the color of light that is responsible for phototropic response. We have these photoreceptors that detect the blue light and initiate phototropic responses. They are known as phototropins. The phototropins trigger the signal transduction cascades that results in at least 2 other responses. Given to its importance to photosynthesis, it is not surprising that blue light triggers an array of responses. This is what I have said. Chloroplast movements inside leaf cells. These movements put chloroplast in positions to optimize light absorption. Means that in highlight, the chloroplast move to the sides of cells to shade each other. And if in low light, they spread out. So what happens if there is a phototropins? There is a movement of chloroplast inside a cell. The opening of stomata, as a result, carbon dioxide can diffuse into cells as blue light triggers the photosynthesis. So again, the responsible for the phototropic response is the blue light. Now let's go to the red and far red light, which talks about the germination, the stem elongation, and flowering. So for us to be guided, the red wavelengths are about 660 to 700 nanometers, and the far red wavelengths are over 700 nanometers. Each of these light, they respond very differently to plants. So we must know what is or what happens when plant is exposed to red light and far red light. What are those things? We have the germination, the etiolation, inhibition of stem and petiole elongation, the chloroplast development, chlorophyll biosynthesis, leaf expansion, branching, flowering, and senescence. So now, I am going to show to you a graph about absorption spectra of pigments. We have the absorption of light in the wavelength. We have the pigments chlorophyll A, chlorophyll B, and beta carotene. Based on the graph, there is a very high absorption on the wavelengths starting from 380 to 500. So from the light, green light, and then also there are some absorption happenings in chlorophyll A and chlorophyll B. The beta carotene is not visible already in the red light. The far wavelengths are not absorbed strongly by photosynthetic pigments, so they tend to pass through leaves. So as you can see, we all know that far red wavelengths is in 700 nanometers and beyond. Based on the graph, you can see there is no absorption of pigments. So since that the far red light do not absorb, the light will just pass through the leaves. We have this switching behavior or photoreversibility that occurs because light absorption make the photoreceptor pigment change shape, like light switching, moving up or down and respond to touch. So now I'm going to show to you an experiment, like how do red light and far red light affect the germination of lettuce seeds. So this one is with experiment. The biologist exposed moistened lettuce seeds, and then these seeds, they are going to be exposed to light, flashes of light that contains one or two wavelengths in sequence. We are going to use two lights here, the red and the far red light. After the exposure to the last flash of light, the seeds were held in the dark for several days. So now we have in the table, we have this light exposure sequence, and then we have also here the germination, the percentage of germination. As you can see here, there's a switching of different kinds of light. We started with none, or the controlled group. The germination is 9% only. And then the next one is red. They exposed the moistened lettuce seeds to red light, and the germination percentage is 98%, which is kind of high. Red and then far red. What happened to the percentage? It becomes 54%, and then red, far red, and then red. The percentage of the germination is 100%, and so on. According to the data, so if you're going to study the data, what is the average germination rate of lettuce seeds that were last exposed to red light? How about the far red light? If you're going to examine the table, if the moistened lettuce seeds are exposed to red light, the last part of theirs of the sequence is red light, the percentage of their germination is high. If the seeds are lastly exposed to far red light, the germination is low. It is almost 40 to 50% lower of that red light. So you have this phytochrome. It is a plant color. It is a special light receptor, different from any of the pigments involved in absorbing light during the photosynthesis or phototropism. So now I have two illustrations here, two different kinds of visual presentation that explains the red light and the far red light behavior. So the red light, it promotes germination. The red light, it causes the phytochrome to change its shape, causing the germination of lettuce seeds. But in the far red light, it inhibits the germination because the PFR is the one that is responsible for the germination of seed. If the PFR will change its shape into PR, the phytochrome PR, it inhibits the process of germination. So by this, from red light and far red light, the red is the one that is responsible for germination. Now, the phytochrome has been shown to play a role in other plant growth responses controlled by light, including circadian rhythm and ethylation. Ethylation describes the phenomenon in which plants have a pale yellow color and lengthen their stems and grow long, thin, and spindly when they are shaded or kept in dark conditions. I have a photo here comparing the normal and the etholated plant. Very visible based on observation. We can see that the etholated plant, they have a color is pale yellow in color, and then they are also longer as compared to the normal plant. Did you know, based on this happened because of the far red light, because these etholated plants are being stored in a dark, or in a place that is dark or in dark conditions. It means that there is no light. The responsible for stem elongation is the far red light. Now let's go to flowering. Again, there are signals that promote the flowering. Phytochrome plays a key role in phenomenon called the photoperiodism. The photoperiodism is very common to all of us. There are three kinds of that involves in this process in the photoperiodism. We have the long day plants, we have the short day plants, and day neutral plants. The long day plants, or the short night plants, bloom in midsummer when days are longest and nights shortest. Long day plants flower only when days are longer than a certain length, usually between 10 and 16 hours depending on the species. Example are radishes, lettuce, spinach, corns, etc. I have a animation here. There are three columns here. Comparison. The full bar is it represents 24 hours. So the line there, since half siya, gives us an idea that it is 12 hours. Then in this bar, you can see two colors. We have gray and yellow. The yellow color represents that this is the light, and then the dark one is the darkness or the night. In this first column, when the plant exceeds to the critical length is if naabot niya ang 12 hours exposure from the sun, it blooms or it flowers. In this second column, the exposure of this plant from sun does not exceed, parang wala nabot ang quota, to 12 hours. It does not bloom. However, in this third column, the plant still flowers even though the minimum requirement to bloom after the 12 hours, there is a flash of light that happens if the plant doesn't, hindi niya naabot yung critical length of blooming. Pero after the 12 hours, if the plant is exposed to a flash of light or artificial light being exposed, the long day plant will bloom. Next is the short day plant. We have the long night plants that bloom in spring, late summer, or fall when days are shortest and nights longest. And the examples of these are asters, chrysanthemum, poicentias, and other short day plants that flower only if days are shorter. You can see here, this is a very opposite one to the long day plants. If the plant it exceeded to the 12 hours exposure to sun, it doesn't bloom. And this second column, if it doesn't reach the 12 hours, they bloom. And then the third one, they didn't reach the 12 hours, but if there's a flash of light, they also do not bloom. So if there's a light, for example, if they do not meet or if they do not exceed it to the 12 hours, and then there is a introduction or a flash of light, they will not bloom. And we also have these day neutral plants of flower without regard to photoperiod. So they are day neutral plants. They don't really care about their exposure to sun if they exceeded to 12 hours or if they do not reach the 12 hours, they will still bloom. Examples of these are roses, snapdragons, dandelions, tomatoes, cucumbers, and many weeds. Clock protein levels rise during the day and triggers the expression of a gene called the constans. So this is pala the responsible, no, the gene. In long day or short night plants, the high level of constans stimulate the production of the flowering hormones. Also, in short day plants, there's also a high level of constans, but it inhibits the production of the flowering hormones. So that made the the experimentalist or the biologist differ about the long day plants and the short day plants. So we're done with different kinds of lights. We have the blue light, the red light, and the far red light. Now let's go to gravity. Gravity or the gravitropic response. We have this famous statolith hypothesis. The leading explanation for how plants sense gravity is based on these two interconnected ideas. Before that, I want you to observe the photo of this one. We have, I think this is a microscopic view of the plant. As you can see, there is a color there like a blackish or somewhat violet. Those are amyloplasts. These amyloplasts are organelles that contain starch granules. The starch is denser than water, and these organelles are pulled to the bottom of root cap cells by the force of gravity. And the gravity, we all know that the direction of gravity is downward. And it is also visible that the amyloplast is going downward following the direction of the gravity. Also, the position of the amyloplast activates sensory proteins located in the plasma membrane. These sensory proteins initiate the gravitropic response. They always stayed on the bottom. We also have this auxin. Auxin as a gravitropic response. So I have this illustration or animation here. There is an auxin distribution. So The first one, this one, there's a normal distribution of auxin since the vertical root is placed vertical direction. No, vertical, so there's a normal distribution. And then the second one is they change the direction from vertical to horizontal, and they are analyzing, or they analyzed what happens to the distribution of the auxin from vertical to horizontal. So what happens during the horizontal, from normal distribution, there is uneven distribution of auxin in the roots. And so now, which part of the root got the higher distribution of auxin? Of course, at the bottom, following the gravity or the gravitational force. And what happens to that if the root is being placed in horizontal for a longer time? What happens is that the roots will bend. The upper part of the root will increase its length, while the bottom part it contracts, making it bend. That's why from very straight one, and then parang there's a pull because that uneven distribution, very large distribution at the bottom, and it will go down and it will bend. So that's what happens. So therefore, they concluded that auxin is a gravitropic signal. Now we also have this the changes in growth patterns. We have the thigmomorphogenesis. In the photo, we have two plants here. If we're going to compare, we have the untouched or the controlled group and the touched. So there is changes in growth patterns. In this experiment, the plants were either touched lightly 2 times per day or left untouched. The flowering stalks of the touched plants were much shorter. And the plants and gravity are not only physical forces that plants sense and respond to. Plants also react to mechanical stresses such as the wind and touch. Now we have the thigmotropism. We are I think very familiar with this concept. Plants respond to touch by moving in a specific direction. This response is called thigmotropism, or the touch bending, and can move moderately fast. We have the tendrils, the one that attaches to a different kind of body. Just like of this one, the tendrils attach to each closer body, which is a metal one. This is the thigmotropism. They are moving in a specific direction. But we have also this non-directional ang ilahang movement. The movement is even faster in touch-sensitive plants. We have this carnivorous plant. The perfect example for this is a Venus flytrap. It captures insect by closing quickly when an insect lands on its leaves. These types of movements occur when a touch receptor cell transduces the mechanical signal to electrical signal. What is this electrical signal? Recall that proton pumps in the plasma membrane of most plant cells give them a negative charge relative to the exterior environment. The separation of charges creates a membrane voltage or membrane potential. If the receptor protein responds to touch by allowing ions to flow across the membrane, which changes the amount of charge on either side, then the membrane potential changes. In this way, the mechanical signal or the touch can be transduced to an electrical signal. The mechanical signal, which is the external stimuli, it will change to electrical signal. That's why specifically the Venus flytrap change or respond. We have this action potential. So the receptor cells at base of hairs trigger the action potential. So inside of the Venus flytrap, we have these hairs. If they are disturbed or touched, then this mechanical touch will be understood as electrical signal. That's why it will respond. In the case of Venus flytrap, it will close. If we're going to compare plants to animals, animals have nervous systems, or they have nerve cells. But what about plants? We all know that plant cells are connected by membrane-lined plasmodesmata. They move more slowly in plants than in animals, but they can still carry information over long distances in a hurry. Now, let's go to the hormones. responsible for growth, for youth, maturity, and aging plants. I'm going to give six hormones, like there are general hormones that are responsible for growth of the plant. We have the auxin, involved in cell elongation and apical dominance, promotes differentiation of xylem and phloem, helps to define long axis of body such as the phototropism and gravitropism responses. First plant hormone that is isolated and characterized, produced in shoot apical meristems and young leaves. So we discussed earlier that the auxin is a gravitropic response. So this one is the responsible for it is the auxin. We have also the cytokinins, which is responsible for budding. It promotes cell division in the presence of auxin, promote chloroplast development, and break lateral bud dormancy. There is a delay of senescence or the aging, and produced in root apical meristems or many other tissues. We have the gibberellins or the GAs. It promotes stem growth via both cell elongation and division. It promotes seed germination. What light? Red light. Produced in apical meristem, immature seeds, and anthers. Those are pollen-producing organs. Abscisic acid or the ABA, it inhibits bud growth and seed germination. It induces closure of stomata and responds to water stress. Act as a stress hormones analogous to cortisol in humans and produced in almost all cells. No, abscisic acid is responsible for the closure of stomata to avoid dehydration of plant. And now we have the ethylene. It exists in gas form. It involved in fruit ripening, induces senescence of fruits, flowers, and leaves, produced in all organs when all plants are under stress. So senescence or aging. The brasinosteroids or the BRs are promote cell elongation in stems and leaves, structurally related to steroid hormones in animals, and produced in almost all tissues. So it is responsible for the body size. So we have this auxin in general. If we're going to summarize it, auxin defines the root shoot axis of the body. The gibberellins trigger the stem growth. Cytokinins promote cell division. ABA slows or the abscisic acid. Abscisic acid slows or prevents the growth. The brasinosteroids increase overall mass, and the ethylene signals the senescence or aging of the plant. The pathogens and herbivores, the defense responses. Plants are constantly threatened by an array of disease-causing viruses, bacteria, and parasitic fungi. In addition, plant roots are susceptible to attacks by nematodes. Nematodes, the soil-dwelling roundworms. Also, just like animals, since plants are living things, they're also vulnerable to pathogens and also herbivores. What they do is they develop this defense responses. An example of some plants that are victim of these some pathogens or herbivores or some climatic actions. The frost injury in soybeans and seedlings, you can see here, the leaves they have somewhat like patches all over the blade. So here the soybean plants dying from sclerotinia infection. So the pathogens are the disease-causing agents. We do not know what kinds of pathogens the healthy plants. So we have these general defense mechanisms, protection of the plants that they already have. We all know that leaves are very shiny. It is a waxy cuticle or the QT that covers the epidermal cells. It is an effective barrier to most viruses, bacteria, fungi, and other diseases-causing agents. And the structure called the thorns that are found in roses, cactus, cacti, spines, and trichomes help protect leaves and stems from damage by the herbivores. Now, we have these secondary metabolites or the plant defense. These are very interesting because it's like a superpower of the plants. So we have these flavorful oils that are found in peppermint, lemon, basil, and sage. They have insect repellent properties. Their aroma are considered as insect repellent properties. And then we have the pitch that oozes from pines and firs, contains a molecule, the pinene, which is toxic to bark beetles. We also have the pyrethroids produced by chrysanthemum plants, which are common ingredients in commercial insecticides. And the tannins, which are ingested by animals. It binds to digestive enzymes and make the herbivores sick. We also have the compounds like opium, caffeine, and cocaine, nicotine, and tetrahydrocannabinol, or the THC, that disrupts the nervous system of plant-eating insects and vertebrates. These secondary metabolites are plant's defense. It's like a superpower of a plant, but these defenses are, you all know that these defenses are effective, but they are expensive for plants because they produce a lot of energy and materials for this to happen. So we have this inducible defenses. It is a response to attack and infections in both plants and animals because they are triggered by the presence of a threat. Now, how do plants sense and respond to pathogens? You have the surveillance proteins. It is signal to plant cell. It is the one that will signal the plant cell that a pathogen is present. The hypersensitive response. The stomatal closure preventing more pathogen cells from entering the plant. The production of toxic molecules targeted to the pathogen, reinforcement of the neighboring plant cell walls to limit the movement of the pathogen, and the rapid suicide of cell in the infected region. In this photo, the hypersensitive response results in small brown spots in leaves. Those cells are dead by suicide that accumulate over the growing season. And by the end of the growing season, it is rare to find leaves lacking these brown spots. Plants that can detect an infection quickly and induce an hypersensitive response remain healthy and show no further signs of disease. The HR, or the hypersensitive response, is similar to cell-mediated immune response in animals, which leads to the death of infected cells. Now, there we have this an alarm hormone that extends the hypersensitive response. We call it the systemic acquired resistance, or the SAR. If you are going to look this illustration, if the pathogen attacks the or the plant, the pathogen molecule, the plant receptor protein, no mag-attach silang duha, the pathogen molecule will attach to the plant receptor protein because it enters. And then then the HR will be triggered, and then we have this systemic acquired resistance that will signal. It's like it will give news to the other parts of the plant. And this systemic acquired resistance, the hormone extends the HR, is called the methyl salicylate, or the MESA. It is the hormone produced at the infection site that travels throughout the body and triggers a slower and more widespread set of events. So the biologists or the experimentalists, they observed that, or they concluded that it is the MESA because the fluid sap leaving the infected sites has elevated levels of MESA. And then the treatments that reduce MESA, reduce number or reduce percentage of MESA, there is a reduce or abolish of systemic acquired resistance. And now we have these proteinase inhibitors. These proteinase inhibitors block the enzymes. These that are found in the mouth and stomachs of animals that are responsible for digesting proteins. When an animal or a mammalian herbivore ingests a large dose of a proteinase inhibitor, the herbivore gets sick. As a result, herbivores learn to detect proteinase inhibitor by taste and avoid the plant tissues containing high concentration of these molecules. So we have this cysteamine. It is the first peptide hormone ever described in plant. I'm going explaining the illustration. When a beetle attacks a potato plant, the damaged cell produces a hormone, the cysteamine, and then this cysteamine will go to the undamaged cell. It binds to the receptor on the membranes of the undamaged cell, and then there are series of reactions that will produce the jasmonic acid. What is this jasmonic acid? Jasmonic acid activates transcription of proteinase inhibitors that will deter the herbivore from further attack. Proteinase inhibitors like that taste bad and make herbivores sick. They really are helping each other. In this way, plants build potent concentrations of insecticides in tissues that are imminent danger of attack. We have also this very amazing. In the 1980s, researchers began to suspect that plants growing near other plants under herbivore attack eavesdrops on these volatiles. When an insect starts munching on leaf, volatile compounds evaporate from the leaf surface and travel through air. In response, they increase their own defenses. So it's like signaling the other trees that, hey, I am being attacked. So there is a release, tensions. In this case, volatiles. No, so it goes to the other part of the plant, like neighboring plants or nearby plants, signaling that they are being attacked. And by this, the other plants, though they are not yet being attacked, they are ready for the possible attack. Now we have these pheromones released from plants' wounds, recruit help from wasps. Caterpillars and other herbivores insects have enemies of their own, often wasps that lay their eggs in the insect's bodies. When wasps' egg hatch inside the caterpillar, the wasp larvae begin eating the host from the inside out. An organism that is free living as an adult but parasitic as larvae such as this wasp is called a parasitoid. Unlike most parasites, parasitoids are similar in size to their host and must continue and kill their host to complete their development. So here in this photo, the caterpillar is a herbivore. It eats the leaves, and when this plant was first attacked by this caterpillar, it produced the pheromones that attracted a female wasp, and then the wasps laid their eggs in the caterpillar. As the result, the wasp larvae grew and developed into pupae and devoured the caterpillar. So, in conclusion, it is very important to know the behavior of plants in terms of their responses as being introduced to different stimuli. By this, we can also discover a lot about the mechanisms of plants and how to address their response. In summary, we have I have a table here showing the different kinds of stimulus by the plants and their receptor, the signal transduction, and their response and their adaptive significance. Ending this presentation, I am going to share to you a statement by Charles Darwin. But a plant on the edge of a desert is said to struggle for life against the drought, though more properly it should be said to be dependent on the moisture. Charles Darwin expresses in his statement about the importance of, instead of thinking negative response, we must think of positive response. It's like having a positive outlook in life. So that is what being emphasized by Charles Darwin. Again, thank you so much for listening. I hope that this discussion made you realize that the plants are also responsive to us. They also attack and they also behave. Well, again, my name is Rael Dialgo Jr., student, Master of Arts in Education in the subject Science 208, Biological Science. God bless everyone.